Assalamualaikum, we are from Group 5. We'll be presenting the workflow of sample collection to the cell processing in Gynae Cytology. Firstly, we will concentrate on the complex procedure of cytology sample collection. One of the most important steps in patient preparation is to explain the procedure to the patient and answer any concerns they may have in order to make them feel comfortable during their examination. The cervix is visible during the speculum examination that follows, making it easier to spot any anomalies or lesions. As we delve further into specimen collection, we examine preparations that are liquid-based as well as conventional. In the conventional approach, endocervical cells are collected using a cytobrush, and endocervical cells are meticulously scraped using a spatula. To ensure a uniform distribution, the collected cells are then carefully put onto a glass slide. On the other hand, when it comes to liquid-based preparations, a broom-like tool called a cytobrush is used to collect the cells. After that, the gathered cells are preserved by being put into a liquid vial. The vial is processed to produce a monolayer of cells on the slide using automated technologies. The slide is then fed and dyed, which improve the visibility of the cellular morphology. A thorough investigation of cellular architecture can be obtained through the subsequent microscopic examination at the end. That's all from me. Thank you.
Next, step number 4 is sample transportation to laboratory. During transportation, ensure that proper fixative is used, which is 95% ethanol. Use appropriate transport container with clear patient information. Maintain suitable temperature condition along the way, and most importantly, prompt delivery is crucial for accurate analysis and interpretation. Then, for step number 5, which is slide staining, also known as pap staining. Pap staining is a polychrome stain that helps differentiate between normal and abnormal cells by highlighting cellular features. It clearly distinguishes between basophilic and acidophilic cell components and obtain a detailed chromatin pattern. Steps in pap staining include hydration, nuclear staining with dehydration, cytoplasmic staining with dehydration, and also clearing. Hydration is a process using a series of decreasing concentration of alcohol to rehydrate the tissue and prepare it for staining. Then in nuclear staining, hematoxylin regressive stain method is applied by overstained slides using Harris hematoxylin. It stains cell nuclei with red to blue after rinsing with tap water or bluing solution, and differentiated with acid alcohol to destain the excess hematoxylin. After that, dehydration takes place using a series of increasing concentration of alcohol to prepare it for counter staining. In counter staining or cytoplasmic staining, it involves orange cysts and eosin azure. OG6 is a monochromatic stain that stains keratin with brilliant orange color. Meanwhile, EF15 is polychromatic stain that contains 3 dyes: eosin, that stains cytoplasm of matured squamous cells; light green, that stains cytoplasm of parabasal, intermediate, and columnar cells; bismuth browns, that stain acid mucin to yellow and stains mast cell granules to brown. Following that is dehydration using 95% alcohol and absolute alcohol to remove excess EF15 and excess water. Lastly, clearing was performed where clearing agent like xylene are used to enhance cellular transparency and visibility.
The next step is the process of mounting, where we mount the slides in dibutylphthalate, polystyrene xylene, DPX, and cover slip. This step is to preserve the specimen during storage, as well as for enhancing imaging quality during microscopy.
The last step of the gynecological sample processing is quality control and slide examination. QC here refers to the internal measures which use a slide for gynecological sample examination. QC has been performed on buccal smear. It has been provided for the cells to be examined by the cytologist and is evaluated for the presence of bulb bodies. The stained slide should be examined starting at magnification times 4 to view adequacy of the specimen, looking at the overall distribution of the cell and marking abnormal cells to be referred, times 10 magnification to look at the distribution and morphology of cells, times 40 magnification to view morphology of cytoplasm or nucleus and confirmation of any infections or cell abnormalities. SLAT screening method must be in linear track from age to age by either vertical method or horizontal method.
So next about the student reflection and experience based on the learning and teaching method in cytology and cytopathology subject. So first, student can understand what is cytology and cytopathology subject. So these subjects are related fields within the pathology that focus on the study of cells to diagnose the disease, particularly cancer and other abnormalities. And next, student also can involve in examining the cells under a microscope. Student can get hand-on experience observing the cellular structure, leading to identify normal and abnormal cell morphology, such as multinucleation, superficial cell, parabasal cell, basal cell, hypochromasia, and perinuclear cytoplasmic vacuolization. Next, student also can learn some methods to detect the cancer, such as gynecological cytology, pap smears that detect the cervical abnormalities, non-gynecological cytology, examining the cells from the other organs like lung, thyroid, or body fluid, and FNAC, fine needle aspiration cytology that collect the cells via a thin needle for diagnosis. And lastly, students also can identify a disease based on the examining the cellular characteristic under the microscope. For example, for the cervical cancer, there are around 18 until 85% squamous cell carcinoma that we can say that the patient have the cervical cancer. And that's all for me about the student reflection experience based on the learning and teaching method in cytology and cytopathology subject.